Hello, thank you all for listening. My name is David Benoit, and today I'm going to be talking about some of the work that I've been doing surrounding multi-species selective fish passage, and more specifically in designing sortable gills for fishes of the Great Lakes. Now, I won't go into too much detail about this, given that this is a special session and there are a number of talks kind of introducing this, but the Great Lakes Fishery Commission is currently developing what is known as Fish Pass. So this is a selective, bidirectional, and volitional fish passage system that has the aim of allowing passage of a greater diversity of desirable species while simultaneously blocking undesirable species. And so if we think about sorting and passage historically, passage designs have typically focused on a single attribute, so often something like jumping or more broadly even swimming performance. And interestingly, these designs typically focused on economically important species such as salmonids. And so you can imagine that focusing on the jumping or swimming performance of a single group might not really allow for the passage of species with different attributes. And so in terms of developing a multi-species passage system, it's thought that passage systems that target multiple attributes are likely to be more effective in passing a greater diversity of species. And so when we think about how we can actually do this and the different attributes that we need to focus on, there are a number of different attribute dimensions that we need to consider. So the first one being phenology and life history. So do the fish actually migrate, and are they likely to encounter a barrier? And if so, at what time of the year might that occur? Next, we can consider morphology. So morphology is strongly tied to swimming performance. So we know that things like caudal morphology, body length, and body shape affect how well a fish can overcome different water velocities. So that's an important consideration. And body morphology can also be used in a sorting capacity by image recognition software that identifies different body shapes. Next, we need to consider the physiology of the species. So in terms of sorting, there are a number of different technologies that can be used to guide or deter fish. So some commonly used ones are auditory cues or electrical stimuli. So in regards to this, we need to look and see whether the fish have the presence or absence of hearing specializations, or the presence or absence of things like ampullary electroreceptors that allow them to detect these stimuli, and that to see if they might be readily, or that we will be able to sort them with these technologies. And lastly, we can look at things like behavior. So where in the water column the fish might be, and in terms of its vertical station when it's approaching a barrier. And also, is it likely to approach the barrier in a group, in school, or is it more likely to arrive as a lone individual? Now, if we think about the great diversity of fishes in the Great Lakes and all of these different attributes that we can look at, you can imagine how creating a passage system that focuses on all of these traits for all of these species might be quite cumbersome. And so one way that we can facilitate this and create a more tractable system is by dividing or partitioning this fish community into meaningful subgroups that we can then focus on. So here I just have an example where we can do this based on things like vertical station, body shape, or trophic level as an example. But you can imagine how if we create meaningful subgroups that we can focus on, we might be able to streamline this design for selective fish passage. And so this leads me to my research objective, which was to group species of the Great Lakes into gills on the basis of their sortable phenological, morphological, physiological, and behavioral attributes. And so this project involved two main steps. First, data acquisition for a large number of species, and then followed by the gill analysis. I won't talk too much about the data acquisition, but as part of this project, we developed the Fish Pass sortable attribute database, which is going to be made available online. And so this database was created using a literature review of peer-reviewed articles, other databases, and government reports and websites. And the database includes 220 species. So that includes native species, established non-native species, as well as potential invaders to the Great Lakes. And so these are essentially species that are considered at high risk of entering the Great Lakes in the near future. The database includes 21 traits. So we have 5 phenology and life history traits, 9 morphology traits, 5 physiology traits, and 2 behavior traits. And so you'll notice right away that there's an uneven spread in the number of traits that span what we call these different attribute dimensions. So in terms of the attributes that we've actually included for phenology and life history, we have migratory status, spatial scale of movement, as well as spawning frequency, season, and temperature. For morphology, we have maximum total length, body depth, eye position and eye size, body shape, different metrics for caudal morphology. So we have aspect ratio and caudal peduncle throttling, which is the ratio between the caudal fin height and the peduncle height. And then we have pectoral fin position and size. For physiology, we have climbing ability, the presence or absence of hearing specializations, lateral line structure, presence or absence of ambulatory electroreceptors, and the trophic level. And while trophic level is not typically considered a physiological trait, it's been shown to be linked to brain morphology. So species with higher trophic levels tend to have a larger olfactory bulb relative to species of lower trophic levels, which tend to have a larger optic tectum, so higher reliance on visual cues. And then lastly, we can look at behavior. So here we can look at vertical station. So again, where in the water column the fish might occur, and then also whether or not the fish exhibits schooling behavior. So given this low number of traits and behavior, we decided to group physiology and behavior for the remaining analyses. So essentially we have these three different data sets, phenology, morphology, and then physiology and behavior. And so what we're left with again are these three different data sets. So spanning those 220 species and the traits within, or the attributes, sorry, within each of these dimensions. But as I mentioned, the data tables differ in the number of traits, but also in the types of traits. So there is a mix of categorical and quantitative attributes. And so one way that we can account for these differences is through a dimension reduction technique, where we identify major gradients in the data through an ordination. And so essentially we can kind of extract major gradients in these axes to account for the fact that we have different types of traits and different numbers of traits. And so to do this, we used a principal coordinates analysis, which is also known as metric multidimensional scaling. So PICO-A summarizes and attempts to represent inter-object dissimilarity in low-dimensional Euclidean space. And the benefit of a PICO-A is that it can handle dissimilarity matrices calculated from mixed data, which is exactly what we have here. And so, as I mentioned before, we can sort of extract this first axis, which is going to demonstrate the important gradients in the data set. And importantly, it's not just a single attribute, but it can be made up of a combination of attributes. And so we can extract that and populate this next data set. Take the second axis for this phenology ordination, and so on. And so we can move on until we create a new data set in this way. Then from this, we can do a cluster analysis and ordination to identify our guilds. And so this approach was first developed by Weinmiller et al. In 2015, not necessarily in a sorting capacity, but in looking at a periodic table of niches. So for our cluster analysis, the first two axes of each ordination were used. We used Ward's minimum variance method, which is a hierarchical approach that aims to minimize the total within cluster sum of squares. And in terms of identifying the optimal number of clusters, we did this through a majority rules framework, looking at over 20 commonly used metrics for determining the number of clusters. And then we also paired this with an ordination. So we did this because cluster analysis can often force a cluster on the data, and we wanted to verify that the cluster structure was actually meaningful. And so we did this here with a principal component analysis, which is another dimension reduction technique that linearly transforms data into a new coordinate system. So for the results, going back to kind of our analytical framework, I'll first show you the results for this phenology ordination. So just to orient you to the plots, each individual dot represents a species, and the closer these dots are together, the more similar the species are in their phenological attributes. The blue diamonds like this represent categorical centroids, so centroids for categorical attributes, so the mean value. And then the gray lines and the gray numbers represent the relationship with quantitative variables. And so the longer the line, the stronger the association in that direction. And so what we can kind of see here is on this first axis, we have a shift from undetermined migratory status to non-migrators to migrators. And on the second axis, we have a change in temperature of spawning. So we have fall and winter spawners, cool water spawners down here. And as we move up to warmer waters, we have summer and spring. So you'll notice that there's a low amount of variation explained on these first two axes. And this is largely driven by the large number of categorical traits. So I won't go through all of these for the sake of time, but we repeated this for morphology and physiology of behavior, created this next data set, and then we did our cluster analysis. So these are the results for the cluster analysis demonstrated through a dendrogram. So again, species that are closer together in this dendrogram are more likely to share attributes. And so another thing to point out is that these black dots represent observed species, so those established non-native species and native species, and the red dots represent potential invaders, so to the Great Lakes. And so we noticed immediately that within each cluster of these different types of species, we have some potential invaders within these groups. Now we can look within a single cluster to get a better idea of what's going on. So just as an example, in this first cluster, we have 36 species. They're typically migratory species of high maximum length that prefer cool waters for spawning, so typically spring spawners. And they're also characterized by the presence of ampullary electroreceptors. And an important thing to note here is that we have, sea lamprey falling out as an observed species. So I just point this out to show that we have, within this cluster of similar species, we have desirable and undesirable species. So we've got walleye and northern pike, which are typically considered desirable, and then we have sea lamprey as well. So we can then compare across the different clusters to see, how each one is characterized. So as we move into clusters 2 and 3, these species are characterized by the presence of hearing specializations, but they differ in their migratory status. So cluster 2, undetermined migratory status, small maximum length, and typically warm water spawners. Whereas in cluster 3, we have migratory species at the medium maximum length, and also spring and summer spawners. Cluster 4, we have migrators and non-migrators of high maximum length. Importantly, though, these are cold water, so fall and winter spawners, so typically salmonids and ciscos. And then cluster 5, we have both migratory and non-migratory species of small max length that spawn in the spring and summer. And so again, we wanted to pair this with an ordination to verify if this cluster structure was actually meaningful. And so these are the results of the ordination in the PCA. So similar to the PCOA, each dot or shape represents an individual species, and it's color-coded to match its cluster. So again, the closer the dots are together, the more similar they are in terms of their attributes. However, in this case, it's not of a single attribute dimension, it's combining phenology, morphology, physiology, and behavior. So in this case, the gray letters here represent categorical centroids, and the bolded words represent quantitative traits. And again, these gray lines or vectors represent the strength of association with these quantitative traits. And so we can see on this first axis that the major gradients are maximum total length, which is strongly correlated with trophic level, and relative eye size in the opposite direction. And so essentially, as we move across the plot, on the left-hand side, we have large individuals, and on the right-hand side, we have small individuals with relatively large eyes. And so we can start to think about how each of these clusters can be used to inform sorting. So for example, in this green cluster, we have large individuals, spring spawners with ampullary electroreceptors, so they may be more susceptible to electric deterrence. In this cluster at the top, where we have fall and winter spawners, a relatively large size, we may be able to use time as an indicator of when these species are going to appear. On the right-hand side, we have undetermined migratory status. So this is an unfortunate one, given that these are a lot of small species that we don't really know a lot of information about. And then this orange and this red species appear to be overlapping. But if we look at them on the first and third axis, they are actually separated. And so this ordination provides good evidence that there is meaningful cluster structure. And again, we can then kind of map on the species that fall within these clusters and think about how we can use this information to inform selective sorting. And instead of focusing on individual species, focus on these 5 individual clusters. So what can we take away from this work? Well, again, we identified 5 clusters based on a combination of phenology, morphology, physiology, and behavior. And we've also identified some of the main gradients in the data set. So this includes migratory status, spawning temperature, maximum total length, eye size, and the ability to perceive sound and electrical fields. And so we can start to use these characteristics that help to differentiate these groups to inform and identify sorting technologies, and also the timing of when these technologies may need to be in place for given species. And so in terms of our next steps, we're hoping to explore the implementation of these guild results at the local level. So this is what we're doing at the Boardman River Fish Pass, which is with a smaller assemblage. And we're also looking to identify techniques to separate species within clusters. So as I mentioned before, even within a cluster of species that generally share a lot of attributes, we have a mix of desirable and undesirable species. And again, this is going to be dependent on our stakeholders, but we need to be able to separate these species that are relatively similar if we want to have a successful selective fish passage design. And that's it. So thank you for listening.